All right, ladies and gentlemen, welcome back. So we're talking about metabolic pathways, right? And it really is all going to come down to consumption versus release of energy. So what we're really talking about here when we're applying those energy requirements to organisms is this idea of bioenergetics, right? Just studying how organisms are capable of managing their energy resources. What do they do to get the energy that they need, and how do they use it? So when we talk about energy in general, it's just the capacity to cause change, or you may have come across it as the ability to perform some sort of work. When we're talking about that in the context of living things, we're talking about the ability for cells to do cellular work, right? And what does that mean? Moving things around, doing cell transport and active transport, to be more specific, moving things across the cell membrane. Um, just being able to drive chemical reactions in cells is going to require energy. So when we look at just some forms of energy, right? We can talk kinetic energy, so the energy that we typically associate with motion, the usage of the energy itself, right? Moving ions with the sodium potassium pump is using kinetic energy, right? Moving organelles around the cells, using the energy to break bonds in atoms so that I can rearrange them. That's kinetic energy. Potential energy is the energy that's stored in something, right? It's stored energy. And it can be based on the location of something, right? So if I look at these divers here, they have more potential energy the higher they are from the surface. And then as they start diving, they start putting that energy into motion. We're converting that potential energy to kinetic. When we're looking at living things, especially in the terms of the metabolism we're going to be talking about here, this unit, we're really talking about chemical energy, right? So chemical energy is stored in the molecular structure. So things like covalent bonds have more stored energy than ionic bonds, which have more stored energy than hydrogen bonds, right? All of those bond types have certain levels of energy stored up, bound up in those bonds. And we can dive even deeper into looking at, is it a carbon to carbon bond? Is it a carbon to hydrogen bond? Is it, whatever the case might be, those also have their own associated energy. Energy is constantly being converted from one form to another, right? So in the case of those divers back in the picture again, we're converting the potential energy into kinetic as that person falls. And so we are changing the energy as we go. So when it comes to an organism's metabolism, it's constantly about transforming matter and energy, right? Transforming chemicals from one compound to another, releasing energy potentially as a result. Um, Releasing heat. Heat, thermal energy, is a form of kinetic energy, okay? But all of this is subject to the laws of thermodynamics, okay? So thermodynamics is simply the study of energy transformations. How do I change energy from one form to the next? And what happens as that energy transformation occurs? So when we look at thermodynamics, and we study this from a physics or a chemistry standpoint, we typically talk about whether or not it's occurring in a closed system versus an open system. And that's going to really dictate what occurs here. So a closed system is a system that would be completely isolated from its surroundings. The more closed off the system, the more efficient it tends to be in that energy transformation. So something like heating up your hot chocolate, but then storing in a thermos. That thermos, for the most part, is a closed system, meaning that that heat should get trapped in the molecules once I put them in there, and that hot chocolate should stay cold. Now, is it a perfectly closed system? Usually not, right? And it will cool off and lose that heat energy to its environment over time. But for the purposes of our explanation here, we're going to consider it a closed system, right? Or at least an example of one. In an open system, that energy and that matter are transferred between the system and its surroundings. We are open systems, right? We are constantly consuming things from our environment, converting them, but the conversion isn't 100%. It isn't perfectly efficient. And so we lose a lot of our conversions to things like heat or other waste products that we end up not being able to utilize and get lost to the environment. The carbon dioxide we breathe out, the water vapor coming out of our mouth, right? So on and so forth. So when we look at thermodynamics, there's a handful of laws that can be applied here to the study of bioenergetics in organisms. And the first law is probably one you may have come across before, and it's simply to state that energy can be transferred, it can be changed in form, but I can't create it or destroy it. So the first law of thermodynamics really is this conservation of energy. You may have come across the conservation of matter, and it's a very similar idea. I can't create or destroy matter, but I also can't create or destroy energy. All I can do is transfer it from one place to another and potentially change the form that it's in, potential to kinetic. Second law might be one that's a little newer to you. And this is really just stating that every energy transfer or transformation that occurs increases this idea of entropy or disorder in the universe. So if we take a look at this, right, living systems increase that entropy in the universe, increase this disorder. It's going to require the input of energy to restore or to maintain that order. So go back to thinking about cell transport, right? Everything, every molecule ultimately wants to go from where it's highly concentrated and spread out as far as it can to where it's lowest in concentration, right? So those molecules bouncing off of each other when they're really tightly close together because they're highly concentrated are pushing each other out, forcing that diffusion or that osmosis or whatever the case might be till everything spreads out. That's increasing the entropy, increasing the disorder as that concentration gradient becomes equalized. Once that equalization occurs, there's not enough energy to force those molecules any further. Okay? If I need as a cell to maintain that concentration gradient, right, like in the case of neurons needing to maintain that sodium and potassium gradient on either side of its cell membrane so that it can send signals, well, what did we need in order to make that happen? We needed that sodium-potassium pump. That sodium-potassium pump required the input of ATP in order to keep the gradient in place. Another way to think about it, if you just constantly move around, do things in your house, in your bedroom, and you never pick anything up, you never expend any energy, eventually that place is gonna be a disaster, right? It's gonna be in full disorder. You're gonna have clothes everywhere, the bed's unmade. All right. If you're a snacker, maybe you've got empty cups and dishes all over the place, right? Um, my son, toys everywhere, right? My house would be an absolute disaster because nobody's expending the energy to put it back, right? Nobody's expending the energy to restore the order. But you go cleaning all of that up, it takes time, it takes energy from you to do that, right? But if you're going to restore the order, you're going to decrease the entropy in this case, it's going to take energy. So living things are constantly converting forms of energy. They're not perfect systems. Remember, we're open systems. And so the conversion of that energy often results in some of the energy being utilized, but a lot of it being lost as heat. All right, lost as thermal energy. And that's going to be the case in any system. But the more closed off the system is, the more capacity to hold on to that heat and be able to use it. When we're talking about living things, a lot of that isn't capable of being trapped, and so we lose it to the environment. Any spontaneous processes are going to occur without much energy input at all. All right, so go back to that idea of um, endergonic versus exergonic, right? Catabolic versus anabolic. Most of those catabolic pathways, because by definition they release energy, are endergonic. I'm not putting in excess energy to make them work, and so these guys will tend to happen relatively quickly. If we're talking about endergonic reactions, right? Exergonic is going to release, exergonic is going to happen relatively quickly. They tend to be spontaneous catabolic pathways, right? The anabolic pathways, the endergonic ones, are going to be ones that are going to happen relatively slowly. And so those are ones when we're talking about what that's doing in a living system often require the enzymes to help speed them up. They need a catalyst in order to do this. Any process that occurs without energy input, aka the catabolic pathways, the exergonic pathways, are going to increase the entropy of the universe. Those anabolic pathways, those building pathways, those endergonic reactions are going to decrease the entropy, but I'm going to require energy to do it. So when cells create those ordered structures from less ordered materials, they have to be able to A, utilize the energy, so that energy has to come from somewhere. They also end up having to replace ordered forms of matter and energy from less ordered forms. That's where your metabolism comes in, right? So being able to take in materials from your environment, create things out of it, is going to mean that because you are an open system, because living things are open systems, we constantly need to be feeding that system with both material and with energy. So that's the whole point to the flow of energy in an ecosystem. When we look at those food chains, right, there's a finite amount of trophic levels we can have because the energy is finite, right? We constantly have to be feeding the system, which means I need lots and lots of primary producers on the bottom of that food chain in order for enough material and enough energy to make it to those top predators for that energy and matter to exist in a concentrated or high enough level for those top predators to have what they need, because they're going to constantly need to be fed, right? So I'm going to give you some basic introduction to this idea of free energy. We're going to talk a little bit about the math. Um, don't get lost in it, but it's definitely something that you will probably come across either in college chemistry classes, college biology classes, so I won't introduce it here to you. But as far as our purposes go, we're not going to focus on it. Um, so let's take a look at it, right? So we're talking about, when we're talking about these laws of thermodynamics and the fact that every system, living systems included, are bound by those laws, means that that free energy change of a reaction tells us how that reaction is going to occur, right? Do I have a release of excess energy, or am I going to need more energy to put it in? And when we talk about living systems, that free energy is the energy that I can use to do cellular work, right? Think about it in terms of cash flow. Do I have excess ATP, excess money to go spend and do things? Well, that's going to take work. It's going to take the input of that cash from somewhere to do those things, right? We are also going to assume in living things that temperature and pressure are uniform. Um, if and when you study this further in, say, like a chemistry course, you will be able to take into account temperature and pressure as well. All right, you increase the temperature, you increase the pressure. Those also play a role in the amount of energy that you have in order to do some sort of work. Okay, so just be aware of that. Living things, we're going to assume your temperature should stay constant, your pressure should stay constant, because we should be maintaining homeostasis. Okay, so a couple of variables here, right? So free energy, just in general, is this variable called G. For the most part, that's not as useful as talking about the change in free energy. So if you've ever seen that little triangle, it's a delta sign, and that always refers to change in. So a delta G is a change in free energy. When we relate that free energy to biological processes, we want to look at the differences between enthalpy change, which is a delta H, along with the change in entropy, so the disorder, the change in delta S, as well as temperature, but we're going to consider temperature in Kelvin. All right, so not Celsius, not Fahrenheit, but Kelvin. So remember, we go all the way down to negative 273, I think, if I got that number right off the top of my head. Celsius is 0 Kelvin. All right, so we're just working on a different scale here. So organisms are constantly living at the expense of that free energy. Okay, so if I look at that free energy, it really is the measure overall of the system's instability, right? How much are these molecules moving apart? How much potential energy am I converting to kinetic energy as I'm going through chemical reactions or moving things, so on and so forth. All right. The tendency to change to more stable states, right? So the more free energy I have, the more unstable the system. But be careful with that because that instability is actually a good thing. It means I have more capacity to do something. Okay, so right here, this is a really big piece to this, right? The more free energy that I have, yes, the system, I should say, is technically less stable, but it means I have a greater capacity to do something with it. Okay, so when I look at molecules that are in high concentration, right, they're going to want to increase that enthalpy, right, increase that disorder. When I increase that disorder, that means I'm lowering my free energy. I'm creating a more stable system, right? I'm reaching that idea of equilibrium, but reaching equilibrium means less capacity to do work, okay? Same thing in chemical reactions, right? Bigger molecules, all that energy is bound up. It's less stable, but it has more capacity to do something. We go through chemical reactions and build smaller compounds out of it, right? We go through a catabolic reaction, and I have more stable system, but less capacity to do work on this end. All right. If I look at a system that's in equilibrium, right, I have maximized its stability, but I have completely eliminated the ability for any energy to exist to do work. Okay. So here are those terms I've kind of been throwing around, that idea of exergonic versus endergonic systems. Okay, chemical reactions. Pathways, even if we want to look at the entire pathway itself. So when I look at these, here's my free energy, right? How much do I have versus the progress of the reaction itself. So when I look at an exergonic reaction, my overall change in free energy is going to be negative. I'm going to lose free energy as a result of this, all right? Because that energy is going to ultimately get released. So my reactants have a really high free energy. My products have a much lower one. The overall net releases extra energy. These tend to be spontaneous reactions. All right. That release of extra energy, well, hopefully that means I can use it somewhere else. If it's a relatively efficient system, maybe I'm making ATP out of it. I can go use that ATP somewhere else. So I basically bind that energy into another molecule. But regardless, this will always end up losing energy to the environment to some degree as heat. Your endergonic means that there's a change in energy that's positive. These are non-spontaneous reactions. All right, these are going to require the input of energy. the free energy bound up in those reactants is lower than the energy that's going to ultimately end up bound in those products. So I have to put in energy from something else. They've got to absorb that energy from its surroundings. Whether it's light energy coming in from, say, the process of photosynthesis, or I'm using energy from an exergonic reaction to drive an endergonic one in the sense of something like cellular respiration. So again, reactions in those closed systems eventually reach equilibrium. So while it sounds nice to have a closed system because I shouldn't be losing energy anywhere, it stays within the system. The problem is that I'm not feeding the system. And so as a result, eventually that closed system reaches equilibrium and the capacity to do anything with it is done. All right. If I continue to feed that closed system, then I can keep it going and it tends to be considerably more efficient. Cells in our body, however, right, are an open system. So not only do we need to keep feeding it, but it then feeds multiple systems, right? And so I can keep that energy moving. I can keep that flow in and out, and I prevent the system from ever reaching equilibrium in that respect. But it's going to require multiple pathways in order to do that.
Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about bioenergetics. Bioenergetics is basically how living systems make use of free energy. And so this plant right here is called Arabidopsis, and basically to the right of it is a diagram showing the metabolism of just the Krebs cycle in Arabidopsis. So basically, this is how it's converting acetyl-CoA into carbon dioxide and then making NAD and fat. And so this is incredibly complex. Each of the red dots represents an enzyme, and each of the lines represents chemicals moving between those enzymes. And so you can see that it's incredibly complex, but you need to know the basics of bioenergetics. And I could sum it down to this. Life requires free energy or available energy. In other words, right now as you think, your brain is cashing in ATP. And if you don't do that, then you will die. So the opposite of using free energy is death. And so basically there are two laws of thermodynamics. First law of thermodynamics you're probably familiar with. It's basically the law of conservation of energy. In other words, energy is converted from sun to wheat to plants to ATP to ADP using that energy in your brain. And so basically the total amount of energy in a closed system is going to be constant. In other words, we can neither create nor destroy energy at each step. We're simply converting it. All energy will eventually end up as a lowest form of energy, which is heat. People are usually not confused by that, but are sometimes confused by the second law of thermodynamics. And basically what that is that as we move through time with each chemical reaction, we're increasing the entropy or the randomness of the universe. In other words, with each chemical reaction from the sun to the ATP in your brain, we're losing order, or it's becoming disordered. Now, what seems to run counter that is the idea of evolution. The evolution seems to make things that are more and more complex over time. And some people point to this as a way that we're violating somehow the second law of thermodynamics, which would be true if evolution occurred over the whole universe. But remember, we're simply one part of the universe. And so we can increase order in one area by decreasing the order of the universe around it. And so that's basically the second law of thermodynamics. Next, we come to the idea of Gibbs free energy. Some kids are confused by Gibbs free energy, so we're going to step through it kind of in a cartoon method for talking about Gibbs free energy. This equation is sometimes what confuses you. And so basically the important thing to understand is what happens with a change in Gibbs free energy or change in free energy. And so we've got three terms that we're going to kind of keep track of. H stands for the total energy, T stands for temperature, and S stands for entropy. And so basically the first thing that you should think about is that free energy, a better way to think of that is available energy. And so what we're going to do is go through three spontaneous reactions. In other words, reactions that occur on their own. And we're going to look at these values of H, T, and S. So let's start with the first one. This is a ball at the top of a slide. Think about this. Does it have more energy at the top of the slide or at the bottom? And the right answer is going to be at the top of the slide. It's going to have more potential energy or energy available to do work. And so let's look at what happens to the H value. So the H value is going to go down in a spontaneous reaction. Let's go to the next example. Let's say that we have a number of molecules that are in a closed container, and think of these as gas. And so the gas molecules are moving around. And let's say I remove one of the walls inside this container. Well, since they're randomly moving around, they're going to spread out, covering that area. That's going to increase the entropy, or S is going up in this case. We're becoming more and more random. Let's say we remove the whole container. What's going to happen? Well, S is going to increase as well. So again, in a spontaneous reaction, the amount of entropy is going to increase. Now let's look at the last one. Let's say we have this bomb here. Is that spontaneously going to explode? No. But let's say we increase the temperature. What's going to happen then? Well, now we're going to have that spontaneous reaction. And so again, this is kind of cartoon look at Gibbs free energy, but let's just let's kind of summarize what we've learned. So in a spontaneous reaction, delta G is measured in this case. So delta H, T, and S. And so looking back to the first one, in this spontaneous reaction, what happened to delta G? In this case, the delta G actually went down. So delta G went down. So what should that do to the change in G? That should decrease it. Well, let's look at the next example with the molecules as we spread out. What happened to those? Those ones increase. What else can increase a spontaneous reaction and increase in the temperature? And so thinking mathematically, if you increase these two values and we're subtracting it from the total energy, what's going to happen to our delta G? Our delta G is going to be negative. And so that's a lot of stuff, but here's the summary. Basically, if we ever have delta G, and delta, remember, change in free energy is less than 0, that's a spontaneous reaction. If delta G is ever positive, that's going to be spontaneous in the opposite direction, and so non-spontaneous. Lots of times we refer to this as an exergonic reaction and this as an endergonic reaction. And if delta G, or the change in free energy, is 0, that means we're at equilibrium. So we've talked a lot about what seems like physics, but we haven't talked about biology. And so let's get to biology. Let's say we have a molecule here of glucose in the presence of oxygen. Is that an exergonic, or energy releasing, or endergonic, an energy taking reaction? Well, that's going to be an exergonic reaction. It's releasing energy as we move from glucose and oxygen to now carbon dioxide and water. If we look down here at the delta G value is going to be negative 686 kilocalories per mole. What does that mean? If you take a mole of glucose, you're going to release that much energy. And so it's giving off energy. So our change in G, or change in free energy, is negative. Where was that energy? The energy is actually in the hydrogen here, because the hydrogen has a lot of potential energy. And as it falls, watch the hydrogen, it all grabs onto oxygen. So we're losing that energy, and we can release that energy. Now let's look at an energy diagram. Well, basically that glucose and oxygen have a certain amount of free energy over here on the side, and when it's done, they're going to have less energy. And so if I were to draw the energy diagram, it's basically going to go up, and then it's going to go down. So this is what an exergonic, or energy releasing reaction looks like. Now, if you know anything about chemistry, you know what this is up here. This up here is called the energy of activation. In other words, you know that just sugar sitting on your table doesn't spontaneously break into water and carbon dioxide and giving off, fire. Basically, you have to put a little bit of energy into it to loosen those bonds, and then it's spontaneously going to break down. But if you look at that the net change from delta G here, G, or free energy here, to free energy here, you can see that there's a decrease in that. And so the delta G is going to be less than 0. That's in an exergonic reaction. If we look at the next one, this is photosynthesis, which even though chemically it's not the exact same steps, it's doing the opposite. We're going from carbon dioxide and water with the energy of light, and now we're making glucose. And so if you look down here at the delta G, now it's a positive value. And so that's an endergonic reaction. It requires energy. If we look at the energy reaction, or the diagram for that, basically we have less energy than we do at the end. And so what's happening? Well, we still have activation energy, but if we look at the delta G, it's going from a small value to a greater value, and now we have a delta G it's actually greater than 0. So this is an endergonic reaction. So in a plant, where does this energy come from? This energy comes from light. And so light is providing the energy to actually boost it to a higher free energy. Now, why are plants doing that? Remember, they're doing that so they can do cellular respiration and then release all that energy in the form of ATP. They're not making that sugar for us. They're making it so they can have a storable amount, glucose, and then they can break it down in doing cellular respiration and then produce energy in the form of ATP. Speaking of which, this is ATP. So this is adenosine triphosphate. This last phosphate, remember, can unattach. And so if we do that, what's happened to our delta G? It's negative. You can see that it's a lot less than it was with cellular respiration, and that's because it's a smaller molecule. If we go back to ATP from ADP, that's going to be a positive delta G. If we break the phosphate, negative. If we form the phosphate, that's going to be positive. And so constantly inside my body, as I move my muscles, I'm breaking that ATP down to ADP to provide energy, and then I'm making it again through cellular respiration. And so what's important when we're talking about bioenergetics? Well, there are two big chemical processes that are super important. First one is photosynthesis. This occurs in the autotrophs, or the photoautotrophs. Basically what they're doing is taking the energy of light, and then they're using that to make glucose. And then secondly, they're taking that glucose through glycolysis, Krebs cycle, electron transport chain, and then releasing that in the form of ATP. In the next two podcasts, I'm going to talk specifically about photosynthesis and how that occurs, and then cellular respiration and how that occurs. But remember, the reactants of one are the products of the other and vice versa. And so the delta G is going to be the same. Negative in cellular respiration, positive in photosynthesis, and I hope that's helpful.